You're listening to Antiviral Frontiers with your host, Rick Kiessig. In this episode, we're talking with microbiologist William Colton about the challenges of antibiotic resistance and his exciting work that combines viral phage therapy and hydrogel technology for drug delivery.
Hi, welcome to Antiviral Frontiers. I'm Rick Kiessig, your host, and our guest today is William Colton. William, would you like to introduce yourself?
Hi, my name is William Colton. I'm a microbiologist by training and an entrepreneur by passion. My background's in antibiotic resistance and kind of novel therapeutics to help treat this upcoming problem that we're dealing with now in healthcare systems.
Great. Yeah, maybe you could tell us a little bit about your phage-oriented product and kind of how it works and how you got into it.
Absolutely. So bacteriophage or phage, kind of the short nomener for it, are these naturally occurring viruses that target and infect prokaryotes like bacteria. And essentially, every living organism has a complementary virus or group of viruses that tends to infect it. And this is our way of naturally harnessing these antibacterial viruses that are very specialized. These aren't like coronavirus or viruses that infect human cells. They're modified to just really target their host bacterium. And they're actually part of our microbiome. So they naturally live among us, around us all the time. We're always exposed to them. So we're essentially harnessing this natural biocontrol mechanism to then use this in healthcare to really regulate these really problematic pathogens.
Right. So, I understand that your product is encapsulated in a gel of some form. How does that work?
Yeah, so our innovation really isn't the phage therapeutic aspect. It's enabling those phage therapies to be easier to use in a day-to-day healthcare setting.
Okay.
So essentially our technology is a hydrogel drug delivery matrix. So think of it as like a webbing or a mesh of these different polymers that we use in a combination to control and protect those phage from host immune response, antibody neutralization, all these kind of hurdles that we've been able to see through early preclinical and clinical trials with other phage therapeutics that we want to be able to overcome to really allow this to be an efficacious treatment for patients. Our technology also enables it to be prophylactic. So not only can we treat active infections, but using this drug delivery technology, we can pre-coat medical devices or pre-treat sites like burn sites where those patients have a high risk of infection and essentially protect those patients from these superbugs that are out there.
Okay. How does that work? How does a coating of a gel protect a wound from infection?
Absolutely. So hydrogels have natural biocompatibility with wound beds and wound matrices.
Okay.
We have hydrogels essentially as our extra polymeric matrices that hold together our tissues and such. So having a complementary structure going along those kind of wound sites provides moisture, structural support, and actively releases beneficial components that promote wound healing, prevent fibrosis and scar tissue formation, which is the bane of a lot of healing processes. You don't want scar tissue formation. That's failed tissue, it's not as viable, and ultimately can lead to ischemia, poor blood flow, and poor future healing and prospects for that patient. And then being able to use our hydrogel to prevent these infections is huge. Once a patient has a bacterial infection in a wound bed, that procedure, the debridement that comes with that, and other issues like risks to sepsis becomes so much more concerning for that patient, and outcomes become a lot worse. So The advantages to using a hydrogel is this biocompatibility, this controlled delivery, and this familiarity for that patient's body and their homeostatic environment that essentially just supports a very easy to implement method. And we use hydrogels throughout medicine nowadays. It's not some kind of novel delivery system. Yeah. We're building off of something that's familiar and comfortable for patients.
Right. And how did you settle on hydrogels as opposed to liposomal encapsulation or something like that? So you're using a sodium alginate?
Alginate is part of the blend. Pectin. We use a blend of pectin, hyaluronic acid, and then some other key polymers that are incorporated that are proprietary. And we change that blend based on the indicated use. So the blend we'd use for a urinary catheter coating is going to be different than what we'd use for a burn wound. But it's the same basic polymers, just in different ratios. That allows us to control the delivery release mechanism, how much moisture is being held, the swelling, all those key components that we have to take in consideration. But why a hydrogel versus these other delivery mechanisms like liposomes that we can use for these applications? Well, we wanted this to be cost-effective, easy to scale, and something that can be implemented throughout the world, including the global south. So we've looked at applications for like XDR tuberculosis, which is a huge issue both in the US as a risk to exposure. There's a really high rate of latent TB in California right now that we're keeping track of. So there's a huge concern about when does this next kind of hurdle happen where these drug-resistant bacteria move out of latency, start causing large infections. We just had one of the largest tuberculosis outbreaks in Kansas recently in U.S. history. So it's a growing concern, and we're watching kind of this issue bubble up across the world. So being able to provide an affordable therapeutic that can be used as a platform across all these different bacterial infections, that was the goal. And we couldn't get that kind of cost efficiency with liposomes. It might be better for intracellular delivery, and maybe down the line, as we're looking at phage therapeutics for intracellular pathogens, like some STIs and stuff, that might be the key differentiator. But for what we're dealing with, these kind of wound beds, medical device associated infections, those are mostly extracellular. So we can really handle that using an affordable basic technique across the board.
I see. And do the hydrogels also let you control things like localization, stabilization, those sorts of issues?
So one of the key advantages that we have now is that these phage that we use in a cocktail, typically we're not using a single phage to treat these infections. We want to make sure we have a cocktail. That way we're not developing resistance, and we're accommodating for a wide variety of bacteria. Phage are very specialized. So if you want to be able to treat or even prevent infections, you need to pool together a group of really strong therapeutic phage to then address that problem. And those phage might have different stability requirements, temperatures that they're comfortable with. So this platform enables all of those phage to come together and be stored without using cold chain shipping. Or phage are stable at room temperature for up to 10 years. So that's the key differentiator, is that we're able to keep these variety of bacteriophage stable and viable until they're needed. And essentially they're rehydrated. It's lyophilized, freeze-dried.
I see.
So same thing with, what are they called? Those little, the ice creams, like the space age ice cream kind of stuff. Same technology, a little bit more advanced. You know, we don't just use basic sugars. We use a lot of different cryopreserving like components in that. But you get this kind of dry, foamy, crystalline structure that, as long as that's not rehydrated, can stay stable at room temperature for extended periods of time. And that's gotten a lot of attention from the DoD for use in austere conditions. We've engaged with a retired general. Now we're looking at collaborations with Henry M. Jackson and General Carvello. So things are really progressing well in that it's recognized this kind of like platform that enables these sensitive biologics to be used in areas where you can't have access to negative 80 degrees Celsius freezer, you know?
Yeah. That's something we have to deal with in our line. We're developing a line of a family of broad spectrum antiviral therapeutics, and our compounds are proteins, and lyophilization is certainly something that we're considering, as well as encapsulation technologies like liposomes.
Yeah, and so our encapsulation
as well.
Is just hydrogel based. It's like alginate. We kept it as simple as possible because, again, these phage mostly conformationally stable. Again, they have a capsid, they've got a tail region. So what we're just trying to do is make sure they're stable for an extended period of time. Being able to build on that, though, and expand into other biologics is the goal with this platform, because we see so much more to this, and enabling like platelet-derived growth factor and these other key biologics that we haven't been able to really implement in healthcare as much, to become new tools that we can use for patients and really improve patient outcomes. And it makes it more affordable. You're not having this high turnover because it's expiring so quickly.
Right. How do you choose the particular phages that you're going to use to target a particular infection? Because they're pretty specific, aren't they, to to bacteria?
And that's one of the concerns. Some people use engineered bacteriophage. Locus Biosciences, for example, uses CRISPR-engineered phage, where essentially they become molecular blenders. When they infect their prokaryotic hosts, bacteria don't have a nucleus. There's no bubble protecting their genome, and CRISPR makes those cuts. So once that phage infects that host, that engineered phage with that CRISPR sequence essentially blends the inside of that bacteria, super high lethality. So the way we select, though, we don't use engineered phage. We use naturally occurring phage. We isolate from the environment in our biobanking process. And that's mainly to help validate our hydrogel platform. But those phage are isolated from clinical isolates, around 100 clinical isolates for each bacterial species we target, that represent what's circulating within the healthcare system currently. So you look at bacterial isolates that come from clinical patients that have backgrounds that represent what we're trying to treat. So for catheter-associated UTIs, we get UTI isolates from healthcare systems and biobanks across the US.
Okay.
that represent what is currently circulating through those symptoms, or excuse me,
Yeah.
those systems. So we take like an epidemiological stance, and then we look
Okay.
at what phage combination has enough range and lytic capacity against those 100 isolates, and then that's how we design those cocktails. So that's how
I
we've been
see.
able to implement essentially a natural selection process to combine overlapping phage so we get enough coverage that, hey, we could use this in a prophylactic sense and essentially prevent these bacteria from being able to grow. Another key advantage with cocktails is these phage can be recombinant. They can swap genes. So if they co-infect, you might end up getting even more genetically fit phage with broader spectrum or certain components from each of the parental phage that are then passed down to those progeny because they're constantly producing novel phage as they replicate. So you're getting
Yeah.
better and better phage even while the therapeutic is working, which is really incredible, but it's just how these viruses work, you know?
Yeah. Yeah. Very interesting. Very interesting. What's your regulatory journey been like for this?
Yeah, so there's a lot of hurdles with that, with phage therapeutics. There's
I can
no
imagine.
FDA approved phage therapeutic out there. There's been some
Yeah.
in like ag and like food microbiology, but even that's been sparse because again, they haven't been able to crack that stabilization and affordability scale. And there's not the ROI that you get with therapeutics, you know?
Yeah.
So it's really difficult to navigate. We're starting to see more and more phage therapeutics approach phase 2 and phase 3 with strong clinical data. And really, once those break through, that essentially opens the floodgates for us and all the other phage therapeutics out there. We're advanced
Yeah.
preclinical. We're working on animal
Okay.
trials. We're approaching
Okay.
phase 1 in collaboration with Mayo Clinic. We're extremely lucky to be working with Dr. Gina Sa, who's like one of the few clinicians that have actually used phage therapeutics to treat patients and save them from these drug-resistant infections. It's like her input
Awesome.
has been key in trying to design this in a clinical aspect with patient care being focused around it. But even with all of that, there's still a lot of uncertainty, you know? And the environment in the US is constantly changing. Our FDA system and kind of the stance on biologics is shifting. And so it really depends on how this new administration wants to tackle that. But I hope they look at it with open eyes as a way to expand the arsenal of therapeutics we can use to help save patients, you know? It's
Yeah.
giving
Yeah.
patients more options.
Sure. So as you gear up for phase 1, then are you having to think about GMP and INDs and all that good stuff, all the kind
Yes.
of conventional, like, I mean, it's basically, is it an injectable or can be an injectable? So, I mean, you
Yes.
basically have to go through the same pathway that a drug therapy would. Is that correct?
Yeah, it depends on the primary indication we're looking at. Like,
Okay.
it's a combination of drugs and biologics. So the phage are considered a biologic. The
I
hydrogel
see.
itself is 510K. So if we were delivering like a pre-approved antibiotic, like mupirocin or some other like pre-approved FDA antibiotic, then we could pursue a 510K pathway. But the key differentiator is that we're enabling this platform of therapeutics. So really, it's trying to get that first approval of a phage therapeutic to then build off of. And once we get a phage therapeutic approved, then we can pursue a 510K pathway. That's what we're shooting towards. And that's kind of the signal we've gotten based on the hydrogel component. But again, we're going to have to see how those phase 1 clinical trials go and really what happens in that process. And then finding manufacturing for phage is really difficult because the bacteriophage we use tend to kill the bacteria that they work with all the time in their bioreactors. So like
Right.
our therapeutic is their number one nightmare in a lot of manufacturing sense. They're constantly dealing with phage, messing up their batches. So that's been a work in progress. We found some local American manufacturers and some in Canada as well that are able to make some accommodations. And we've given them some certain SOPs and protocols to really help prevent any crossover events from happening and such.
Okay.
So
Yeah.
that's
Yeah.
been reassuring and they're able to meet the scale that we need them at. But really, it's great because 1 liter of these phage cocktails, one liter, can produce around 43,000 units of therapeutic hydrogel. So
Wow.
that's 43,000 patients that can be treated from a fairly small volume because these phage
Yeah.
grow at such large numbers.
I see. Yeah. Aren't phage like the most abundant biological agent on Earth or something
Yeah,
crazy like
yeah.
that?
Wherever there's bacteria, there's a dozen or more so phage there living alongside.
Yeah.
And like ocean water, one of the greatest sources of bacteriophage. All those little microorganisms living throughout that environment, there's viruses feeding off of all of them and regulating those populations. You can even see it from space with some algae blooms. Yeah.
I think I read somewhere there's something like 10 million phage per drop of seawater or something.
It's some insane
bizarre,
number.
some insane number.
And we deal with them in the lab on something called plaque forming units. So plaques are those kind of clear zones that you get when you grow them on a lawn of bacteria. And so we measure them in plaque forming units, but in log differences. So it's not like 1 or 2 more or 3 more. It's a log 10, log 5, log 7 difference between cultures, depending on how you optimize it. So we're able.
Do you have your own lab?
Yes, we operate a BSL2 R&D lab here. We're actually planning on moving out of this space and into one in Phoenix to work with our collaborators in Mayo Clinic and Arizona State University. So it'll be a big upgrade, moving from kind of a Midwest environment closer to a coastal ecosystem. I've been here for 6 years. I started this company as an undergrad. It's been great growing here, but Oklahoma, it hasn't provided the resources to really develop my company past that point. So we're really looking forward to expanding beyond,
Yeah. Yep. We also have our own called a PC2, but same idea as a BSL2 lab here.
Yeah.
So we've been through that. And we grow E. coli as our express our proteins in E. coli. So
do y'all work with that modified E. coli that's able to do like the disulfide bonds? You're able to use it for like recombinant protein expression and stuff?
Yeah, that's right.
That's awesome. Yeah. That was such a cool innovation at the time because like that was such a limit for science for such a long time, was finding a good model organism to host that. And they were finally able to make the right modifications so we don't have to use yeast anymore for all that stuff.
Yeah. It's been great for us. Very productive platform. How do you handle things like manufacturing and CMC and those sorts of issues? Have you had to face that yet? Or are you still in kind of preclinical?
We're still preclinical. Those aren't things that we necessarily have had to face yet, but it's definitely stuff we have to consider, And the thing is, we can meet batch standards with the kind of phage concentrations we're working with. It's really diluting the phage within the hydrogel matrix to meet the quantity and quality that we need for that certain indication. So typically we work with a MOI, which is like the ratio of phage to the amount of bacteria required to treat, like to indicate an active infection. So we do an MOI of 1. So it should be 1 phage virus particle for just about every single bacterium present for us to really eliminate and clear that infection effectively. So in catheter-associated UTIs, there's around 10 to the 5th colony-forming units. That's around 10,000 bacterial cells per milliliter of urine within that patient when they have an active urinary tract infection. That's the requirement threshold to diagnose. So within our hydrogel matrix, we usually keep around 3 log more. It's around 10 to the 8th to 10 to the 9th plaque-forming units of our bacteriophage within that matrix. So that way when it rehydrates and you're dealing with that large volume within that patient and that phage is diluted over that period of time and you have that extended release, it matches the ratio and you're able to clear that bacteria while still having enough phage left over a period of time that you're not risking reinfection. Because those viruses, once they're released from the hydrogel matrix, are not stable for very long. They're cleared by the host, they're cleared by the system. They don't last well once they're outside of that microenvironment that's stabilized them for so long. So there's pros and cons to that, in that it doesn't expose the patient to as much risk of like a long-acting therapeutic, but you also have to make sure there's a constant release of those phage, which is why we've designed this hydrogel in the way that we have. Because if there's not, you give that bacteria a chance to start growing and then overcoming those bacteriophage. And we can't do that. We don't want to develop resistance on these kind of new therapeutics and the same mistakes we've made in the past.
So do you have to, like for a UTI, for example, do you have to diagnose which bacteria species are there in order to know which phages or which phage mix to use? Or have you been able to come up with some generic mix that's effective against a wide cross-section of common bacteria?
Yeah, so we've been able to target usually the leading 3 to 5 bacterial species that are indicated in whatever infection is implicated for prophylactic use. So in order to prevent the infection, we want to look at coverage that makes this more competitive than like the leading prophylactic alternative. And so for catheter-associated UTIs, it's silver and nitrofurazone-coated catheters. And those are around 40 to 60% effective. So as long as we're able to overcome that threshold and provide a better alternative to that patient by actually able to proactively prevent that infection from those drug-resistant bacteria without the risks posed to that patient, because there's adverse side effects to nitrofurazone and silver coatings. Patients can be allergic, patients can have irritation and site issues that they're not going to have with a hydrogel coating with Phage Therapeutics. So being able to provide that improved threshold and being able to provide 80 to 90% coverage versus 40 to 60% is huge. And that's what we shoot for. So we design the Phage cocktail around usually the 5 leading bacterial strains. Again, 100 clinical isolates that are representative for each bacterial strain and species. And then we build off of that. And then we look at overlap, because sometimes these bacteriophage have enough host range to be able to overlap. So we've seen some bacteriophages that are able to infect E. coli as well as salmonella. And so we're able to get some overlap from these bacteriophages and actually optimize these cocktails, and they're not as large and as problematic as they could be, which is really great.
I see. Interesting. And what about biofilms? Is that have you been able to show efficacy of phages against reducing biofilms?
So biofilms are my background. I studied Pseudomonas aeruginosa biofilm formation as part of like my undergrad studies, and really was kind of the leading reason behind us using phage, because phage naturally produce enzymes that degrade key components of biofilms. Biofilms are also a polymeric matrix, but it's literally just like a shanty town for bacteria. It's their kind of resourcefulness of using whatever they can around them to kind of protect themselves and essentially hibernate. They go into a quiescent state where they're like metabolically inactive, kind of like bacterial sleep. And a lot of the antibiotics we use target those metabolic processes. So while those bacteria are quote unquote sleeping, the drugs we're using and trying to treat with are not effectively killing them. And so phage are able to actually infect these quiescent bacteria, reactivate them and hijack them to be used to produce new phage progeny. So it doesn't matter if the bacteria is sleeping or not, quote unquote, the phage will take advantage of the situation regardless. And at the same time, a lot of papers have come out where they're even able to use these microtunnels within the biofilm, because phage are so small, to disperse themselves throughout the biofilm, essentially eradicate the bacterial populations inside. And we've shown strong efficacy. We've done Congo red binding assays. That's something I even used to do in undergrad, and crystal violet staining assays to look at biofilm reduction. And in some cases, we're seeing 80 to 90% reduction in biofilm. So it's eradicating these biofilm matrices, because that's awesome. That's what the phage have to do in nature, though. You know, biofilms are everywhere. So many bacteria use this, and these little viruses are pros at being able to get in there, infiltrating and getting the job done. It's how they've stuck around for so long, you know?
Very interesting. Have you looked at cancer-related biofilms?
So engineered phage are something that we've been looking at. Some instances we've looked at are like engineered bacteriophage for reactivating latent HIV using modified ligands on the outside of the phage capsid. It uses a T4 bacteriophage, one of like the model bacteriophages, one of the most well-studied ones. And it's actually able to reactivate latent HIV with hidden cells, ones that normally we can't reach to because of the lipophobic properties of a lot of antiviral drugs like that we use. They're not able to reach those deeper cell spaces within lymph tissue, lymphatic tissue, and fatty tissue where there's latent populations of HIV. These phage are able to somehow get into those areas because how small and mobile they are, and they're mostly a protein structure. And they can get in there, bind to those cells, reactivate that latent HIV. And if that patient's on antiretroviral therapies, it essentially clears the system, because those new viral particles from the HIV aren't able to reinfect and cause new cells to become proviruses. And you're just killing off any of those kind of hidden ones. So in the same way, we could maybe look at engineered bacteriophage to target cancer cells throughout the body, depending on what type of receptors they're producing. You can use them in combination with engineered monoclonal antibodies, you know, and maybe that makes it harder for that cancer cell to survive, or easier for our immune system to identify those cancer cells that are normally trying to hide from our innate immune response. So there's a lot of potential, really, with these bacteriophage particles, you know.
So is the hydrogel injectable? Like, can it get to deep tissues,
So
or is it limited to surface applications?
we've looked at blends using something called a poloxamer to allow an injectable version. So poloxamers are these really cool hydrogels. They are kind of reverse of like what you think the intuition is for a hydrogel. Normally you heat up something to melt it, you know? This is the opposite. This solidifies at body temperature and becomes liquid when kept in the refrigerator. So we've been able to lyophilize these poloxamers with our normal hydrogel matrix. It does change kind of the texture and the delivery profile, but we're still able to get a sustained delivery that's needed for therapeutic use. But essentially, it's a cold injectable hydrogel. So when it's resuspended within its ionic buffer after being lyophilized, as long as that ionic buffer is cold enough, it'll stay liquid. But as soon as that's injected, within a matter of 60 seconds, it solidifies at the site of care. So if you have a joint infection, for example, and you need a rapid on-site treatment that needs to be injected at the site of that infection, this poloxamer gel can then be injected into that knee cavity. It's biodegradable, so it naturally degrades over 5 to 7 days within the patient, and would release those phage over that period of time to actively treat and support healing within that area. Because again, it's providing moisture, wound bed support. So whatever necrotic tissue, whatever kind of damage has been done by that infection will then be soothed essentially by this hydrogel that's being introduced.
Okay.
So
that sounds like so like near surface, like a knee, but maybe not like a liver tumor or something.
No, again, those kind of infections. So we could do a localized injectable hydrogel, but that's not really one of the key indications we're looking at right now. We're mainly looking at either respiratory delivery, medical device coatings, or surgical site infections or burn wounds. So these are the key kind of areas. So XDR-TB, we've created a dry powder inhalable hydrogel formulation with bacteriophage that can be used both prophylactically and therapeutically in force preservation for military officials and personnel. So this aerosolized hydrogel can be implemented in cases where there might be a TB outbreak on a military base to essentially protect other military personnel. So really it's more frontline. As this develops, we might be able to do more complex procedures like liver abscesses or deep chronic infections that are much more problematic. But right now, the main focus, probably the deepest that we'll get, is orthopedic infections, hip replacements, the stuff that killed my grandmother when she underwent a hip replacement procedure. Ended up getting a drug-resistant staph infection.
Oh, wow. Awful.
Biofilm formation. They tried to go back in with an antibiotic flush. Couldn't really do much, and they just put her on a central line of vancomycin and morphine for 6 months until she died from sepsis. So it's terrible. But like, I'm not unique. You hear about this stuff all the time in healthcare. So it's a huge problem, and we're trying to do something about it, you know?
Yeah. Drug resistance is a terrible and growing problem.
And we're running out of options. We haven't had a new class of antibiotics since like 1985. It's been a
Yeah, it's bad. Kind of along those lines, have you been able to or have you worked with like emergency use authorizations or expanded access, that sort of thing?
Our collaborators at Mayo Clinic, Dr. Suh has used emergency use authorization in the past to treat patients. And so we're looking at those options. We want to pick the application based on our animal trial data that's best. So once we publish that, usually we're looking at Q1 or Q2 of next year to go public with that animal trial data and use that to help support our Series A raise. But we're doing some parallel studies. And we're trying to look at what's the strongest indicator to pick as our leading use case. And so once we kind of take that data, Dr. Suh will then use that with our preclinical meetings with the FDA and then help design potential clinical applications and trials and potential patient qualifications that we could use for emergency use authorization. There's a lot of risk that comes with that as well. These patients are typically in critical condition. So we really want to be mindful of where the patient is and what's going on. But if we have that opportunity, we would love to take that, to be able to provide that kind of support for that patient. But we want to make sure we put the best product forward if we do end up pursuing an emergency use authorization. Because again, it's a really tough situation for that patient.
Sure. Yeah, you mentioned Series A. We just closed our Series A about a year ago now, so I've been through that. It was quite a wild ride. I'm curious what's your thinking and approach to that fundraising process is and has been.
Yeah, so really kind of taking the platform aspect and showing that our technology has these applications that are licensable, that are able to kind of fail-safe and risk-proof the regulatory pathway. We have parallel applications. A lot of what we can do in humans, we can do in veterinary care, which is also another key market. So periodontal disease, wound infections, surgical site infections, all of those are common issues facing veterinary care. So we actually collaborate with OSU's College of Veterinary Medicine as well, and they help design our animal trials. But we work in tandem to kind of provide these therapeutics to veterinary pharmaceutical companies like Zoetis and Elanco, who are interested in these kind of technologies and have acquired similar ones before in the past. So we're looking at as many options as we can to help maximize value and maximize this kind of IP that we're working with. And that's how we've been able to build an investable narrative. Some VCs have gotten back with us and told us that we need more focus. We want to be able to see a single indication. And that can be really hard when you have a technology that spans a lot of different applications. So we've tried to streamline and focus on, animal trial data, show them what works in vivo, and then with the strong data that we're working with, raise a much larger Series A, bring in our collaborators and their institutions. So Mayo Clinic Ventures is affiliated with Mayo Clinic and is able to invest in affiliated projects. So if we're able to show strong enough data, they could lead our Series A round. We could also use some of the other investors we've been speaking to for a period of time, but the biotech ecosystem in the US is in a whirlwind right now with how grant funding has been upended. It's really, really, really unclear what that future is going to look like. So we're trying our best to provide a path of stability and something that's straightforward to investors that shows that even with adversity, we can provide an ROI. So it's our best attempt.
There's a lot of what you're saying echoes with what we've been through as well with our broad spectrum work. I'm curious about your approach to IP as well. Are you doing like just patents or something broader?
So we filed a PCT internationally, US, EU, Japan, Canada, and Australia. And then we're doing divisional patents as well for each of the applications. So we've got an umbrella that essentially covers the know-how behind blending the hydrogel, the microencapsulation procedure and how that's put into the hydrogel matrix, and then that kind of lyophilization freeze-drying process with cryoprotectants to then kind of wrap everything up and provide a thermostable product. So
I see.
We've been able to build that platform IP, that umbrella patent that kind of protects, but then we have to do divisionals for each of those key applications because those can eventually then be sub-licensed, can be spun out and turned into other applications. But it's expensive. It's extremely expensive going through the IP process. So we have to be strategic about where we allocate those resources. So it's a game of cat and mouse. Once we kind of have a key technology that we know is valuable, we want to slap that patent on it and then have that value established. So, it's part of the process. It's IP strategy. But it's great that we have this opportunity. In case we need to switch to a different indication, a pivot's always available and just steps away. So that's always really great to have.
Right. What are you leaning towards as your initial indication?
So typically catheter-associated UTIs have been our strong suit. It's the number one hospital-acquired infection. They're recurring. It's a 3% risk, compounding risk per day that catheter is left in. We have a aging population in the U.S. That's going to drastically increase for the next couple of years. And so nursing home care, medical care, a lot of those patients end up using urinary catheters. And if it's an indwelling catheter that's left in for up to 30 days, they're almost guaranteed to have an infection by the end of that time. And as more strain comes on healthcare workers in the U.S., as we limit the ability for foreign healthcare workers to come in and work in our system, there's less and less medical professionals there to take care of those patients. And that means there's more and more risk for things to go wrong. And so with those kind of key drivers in the ecosystem, that's where we see a huge impact. We can prevent this, what shouldn't be an issue, but is now one of the leading problems in their healthcare systems. And patients die from this. It starts as a urinary tract infection, splits to the bladder, then the kidneys, and then they're septic. And a lot of times, they've got other comorbidities that make that even more problematic for those patients. So it's one of our key indicators. But again, we're also looking at XDR-TB and other pulmonary applications. My background's in Pseudomonas infections for cystic fibrosis patients, so I always have that in mind. And then we've looked at some other applications as well, STIs as well as C. diff infections. So C. diff is actually caused by clindamycin use and some antibiotics. Because again, antibiotics are like a nuke. They wipe everything out, good or bad. It's indiscriminate. And sometimes that leaves opportunity for drug-resistant bacteria like C. diff to kind of take over. And it's terrible for those patients. It's a single bacterium, so it's one of the easiest low-hanging fruit for us to target for phage therapeutics. It's not polymicrobial. It's pretty genetically distinct. There's not a lot of diversity. So it doesn't take a lot of different phage to then accommodate for that. And with phage therapeutics, you can co-deliver probiotics, which you can't do with antibiotics.
Okay.
Because the phage won't target lactobacilli. They'll target the C. diff.
Right.
You can reintroduce a stable microbiome and eliminate the problematic bacteria in one go, which is really great.
It's fantastic that they're targeted as well. So you're not killing off the entire microbiome like you are with a lot of antibiotics.
And you leave the patient susceptible because the microbiome is essentially like a secondary immune system. They're there to outcompete the problem, which is all around us. And it takes time to cultivate that.
Yeah. How do you avoid off-target effects?
So again, pre-targeted. One of the issues that we can kind of see sometimes is like there's K12 derived strains of E. coli, probiotic E. coli that naturally exist within our microbiome. What we found though is there's a kind of stochastic trade-off between antibiotic resistance and phage susceptibility. The more resistant a bacteria is to antibiotics, typically the more susceptible they are to phage therapeutics and vice versa. So when you have something that's evolved and kind of dedicated this genetic energy, this genetic token, essentially, that they have a limited amount of to antibiotic resistance, producing these enzymes and key countermeasures to antibiotics, that leaves them susceptible to other infections, so like bacteriophage. They don't have as much genetic capacity to then accommodate for that as well. So by using either a combination of phage and antibiotics, or by using phage specifically that target these drug-resistant strains that look at like proton pumps as a phage receptor target type, those are commonly used to help target those are commonly used against like aminoglycoside antibiotics or other antibiotics. Essentially, the bacteria pumps them out of their body. But you can then use those pumps that are used as a countermeasure to antimicrobials as a target for those bacteriophage. So again, it's a consideration you have to take into play. There could be off-target effects, but nothing near what you would see with antibiotics, given that, again, it's a close spectrum. And a lot of the times what's causing these secondary infections in hospital systems are commensal bacteria that are taking advantage of being in a site that they're not supposed to be. So if we're treating a certain site for a certain infection, those bacteria are typically not supposed to be there. So if they're not supposed to be there in the first place, typically we're not going to hit anything that's off target. So there's some coverage and stuff, but key advantages compared towards the antibiotic arsenal we're working with now with molecular antibiotics.
Right. So in your animal testing, like for UTIs and so on, is that a straightforward thing to test for in vivo?
So we use a mouse model. It's standardized. We've been able to kind of base that off of other research that's been used. Again, very difficult with mouse catheters, very small. But again, we've been able to work with the vet school, and that's been really great to get that professional input. Dermal infections and wound care have been much more straightforward. Those are much more standardized for mouse models. Working with drug-resistant staph is also pretty standardized. One thing we have to take in consideration is the bacteria that can cause systematic infections and systemic infections within these animals are not always the same bacteria that can cause those same infections in humans. So we have to be very selective about the strains we work with in those situations. But one of the vets we work with is a veterinary pathologist, so he's able to do the histopath, really do a great selection process at looking at what bacteria are relevant towards a clinical setting, but will represent that infection best within the animal trial and animal study. So I'm really grateful for that because, again, that's not my background. I'm not a veterinarian, so having that support is huge. I know bacteria, but sometimes when it comes to like the zoonotic stuff, it's a little bit outside my field, you know?
Yeah. Yeah. So you've been pleased with the quality of the animal models, at least for this type of work?
Absolutely. And like, there's been a lot of flexibility working with a veterinary school versus like a contract research facility, you know?
I see. Yeah.
They're much more academically minded. So they're able to actually pull from a lot of academia, novel techniques, and they tend to be a little bit more creative, which is really cool. They take on like a perspective that's like, okay, this is more of like a challenge. Let's tackle this versus like, oh, this is going to cost you this much. This is our standard protocol. So it's really nice having that change in pace and being in academia and like being surrounded by that. And like, it's great because students also get to be involved. They always have students coming in through the veterinary school. So they get to have a chance to participate in these animal trials and provide like meaningful support towards something that can make a huge difference in healthcare. So it's like a really cool environment to work in. And they've done a great job so far. So I really appreciate what they've been able to put through.
You're working with a CRO as well?
We've looked at working with CROs, but again, we've been satisfied with the work that we've gotten with OSU's College of Veterinary Medicine. And like, I can't recommend enough looking at collaborators like that. Having institutions like that, they pull in a lot more resources than you would think. And cost is not always like a priority. They're interested in publication. They're interested in novelty. So that adds like an added value on our end because it really helps to up the scientific rigor, you know?
Sure. I'm curious how you draw the line between the work that you do in your own BSL-2 versus what you contract out. This is something that we're always discussing internally about exactly how we do that because, of course, we do similar things. We work with academia and CROs and others to do outside work, and we have our own lab. So, yeah, there's always a bit of a discussion, or often, for us. Just curious if you have the same sort of thing.
Whatever we can do in-house, we try to. It's most cost-effective. It usually moves the fastest, you know? That's great, the great aspect of having our own space. But when it comes to something that we want to be at the highest form of approval, the highest form of representation of our technology, something that's like a kind of a pinnacle, we would then turn to one of our collaborators. So again, if it's outside of our scope, outside of our specialty, and we know that we can't kind of perform to the standard that we need, that's when we outsource it. So animal trials, OSU Vet Med. With Mayo Clinic, we have a sponsored research agreement. So once we've optimized our animal trial data and gone through that process, we can then provide that data to Mayo Clinic and work with their research labs, then advance that, publish that data, and have that kind of seal of approval with this high-level clinical institute supporting this research and pushing it forward. So it's being strategic about really taking the best that we can put together and putting that forward for our collaborators to then push on to the next level. Because we've worked hard to get that point, but again, it's like a snowball. We got to make sure we add enough momentum and kind of force behind it to keep those partners engaged so they have something to move forward with. So again, we're scrappy as well. We went through COVID. We're going through this kind of change in the biotech ecosystem right now. So whatever we can manage to do on an effective budget, we will keep in-house, you know. But if it's absolutely something we have to outsource, whether it's sequencing, which is something we have to do, it's fairly cost effective. So it's not too much of a problem or burden for our biobanking process. And again, phage genomes are small, thank goodness, because otherwise it'd be a ton out of pocket, but they're about as big as some plasmids. So it's pretty straightforward getting that sequencing done. But yeah, most of the stuff we do in-house, some we outsource, but typically we try and get as much taken care of as we can in-house. We do all of our biobanking in-house. We keep all of our isolates in our cryopreserve bank, and we just work off of what we can, you know.
You mentioned biobanking. I'm curious about how you approach that. Is it, as far as I know, there aren't any good biobanks for phages, or are there? Maybe I just haven't seen it.
There's none that are like public, you know. We have like the ATCC, the American Type Culture Collection, which is like the gold standard for getting whatever. And then you have like smaller collections that some universities have, but really there is no great biobanking program. We've got more AI tools for phage than we do biobanking programs for phage therapeutics, which is concerning because like you need access to these actual viruses to really understand, work with, and characterize them. So there needs to be a push for a better centralized bank. There's some in Europe. I know like Poland has done a lot of phage work. The country of Georgia in Eastern Europe has done a lot of great phage work in Tbilisi. They have their own phage research center. From what I understand, Eastern Europe has really engaged a lot with phage therapeutics, especially in the past. When the Iron Curtain fell during World War I and World War II, they kind of kept with the Soviet side and did not have access to penicillin like we did in the West. So they stuck with phage therapeutics and were able to kind of push that a little bit forward. But again, they don't have access to the technology we had today, like lyophilization, back in the late 1800s, early 1900s. So they were doing the best that they could, but again, it wasn't at the point where it could compete with something like penicillin, which was essentially a wonder drug at the time. You know,
is it well known which phages infect which bacteria? Is that mapping well understood across the wide cross section of bacteria?
It depends on the bacteria that's being studied. You know, if you're looking like E. coli, you can pretty accurately map what kind of viruses might infect it based on what we already know and what's been characterized using predictive mapping. A lot of E. coli bacteriophage have been characterized and been sequenced, so those patterns can be matched. There's certain things you can look at within the machinery of the bacteriophage and the target to look at compatibility and make sure one can be expressed within the other. And so that can be used for predictive mapping, but it depends on how well understood that host species is. You've probably seen that they're trying to do in silico full cell modeling, which is almost impossible because we're still discovering new things about the cell every day. And so it's jumping before the gun. And with what we're trying to work with, really you have to do the lab work and grow the phage on the bacteria to know that it does the job. And that's what we do when we do our biobanking. If we're going to preserve a phage isolate, it needs to have therapeutic efficacy. So we will screen those isolates, and once we characterize one that we believe has therapeutic value, we then sequence and store that for long-term use. So, yeah, it's interesting. You've got to use the wet lab space. You can't use everything on sequencing. AI can't replace everything. These are naturally occurring. Like, why do any of that? But there's a lot of cool things going on now too. They're able to use like essentially sequencing to express phage and essentially build them without using a host. So there's a lot of really cool tech that's being developed out there nowadays. It'll be interesting to see where that kind of goes.
Yeah. Interesting. On the getting back to regulatory for a second, have you had to approach things differently? It strikes me that there's maybe I'm mistaken about this, but there's like a sort of semi-topical aspect of it, and then there's like an injectable aspect, and there's a medical device aspect. Do those do they overlap enough to simplify?
No. Again,
FDA process, or do they make things worse?
Like our aerosolized kind of formulations would stay within the same grouping. So an aerosolized Pseudomonas aeruginosa treatment would be in the same kind of grouping as an XDR-TB aerosolized formulation. So it would fall underneath the same kind of regulatory strategy. In the same way, like a medical device coding for a hip implant or for an osteopathic procedure would fall within its own kind of category. And then dermal applications, burn wounds, surgical site infections would fall within a similar regulatory category. So there's subgroupings within this kind of umbrella technology that each have their own considerations when kind of tackling that FDA regulatory pathway. And also considerations we have to take into play with patient recruitment. We have to have enough patients that have these infections to be able to recruit and do a phase 1 clinical trial, phase 2 clinical trial. There's not that many osteopathic infections to pull from. But catheter-associated UTIs are going on all the time. So it's not difficult for us to recruit patients dealing with those infections to be a part of our trial, because this is an ongoing issue. Whereas it's a lot more difficult to find some of these rarer bacterial infections that are just as devastating, but just don't occur enough for us to use for validation.
Sure. Are you planning your clinical trials, your phase 1s in the States, in the U.S.?
Yes. So that'll
overseas, or have you?
No, it'll be in the U.S. We'll work with American-based patient cohorts. We need to look at regionality. We're looking at what facilities we can work with Mayo and how we can recruit patients through Mayo Clinic's facilities. We're also looking to other institutional partners as well. It's a big push to do that. But again, we need to see how this animal trial data works out and what key indicated use case we want to move forward with. So that'll be a huge indicator for how we then proceed with engaging with patients for that cohort.
Okay, right. And for your hydrogels, you mentioned earlier you use ratios of different components. How do you go about developing that? What's your process
Yeah,
look
so it
like
really
there?
is trial and error. Looking at consistency, delivery profile, it's a lot of testing, a lot of elution profiling, and then kind of quantifying what that release profile looks like. So testing different
Hmm.
blend formulations, and then kind of getting an idea of where to start based on published literature and a fundamental understanding of how that works. So looking at similar hydrogel formulations, matrix sizes, what's been characterized, and then building off of that to then develop our own formulations in-house. And that's how we've been able to do it over the past 6 years, is we've been able to take an understanding of the basic hydrogel structures that we can are familiar with, and then blending those together to get different properties that we need from each hydrogel component for optimized phage use within a clinical setting. So making sure it's biocompatible, making sure that it has the right swelling ratios we need for the indicated use, and making sure that the matrix size allows for delivery of the bacteriophage without invasion by macrophages and other dendritic cells that could eliminate bacteriophage within the hydrogel. So a lot of key components
Okay.
that go into consideration when we develop that. And a lot of that can't be predicted. So we have to actually go through the wet lab testing. That's what takes a lot of
Yeah.
time. We started this in May of 2019, opened up the lab in January of 2020, and it's just been iteration after iteration since then, really building out these hydrogel platforms and then specializing them for their indicated use. I can't tell
see.
you how many different versions of formulations we've gone through. Oh my lord!
I could imagine.
It's a lot, and it's just gels. We're just working with gels all the
Yeah.
time, and it's like, how much more gel do I have to work with to be able to get the right formulation needed? But we're able to actually like optimize that, and working through the right ratios, we're able to get delivery profiles that are really great for the therapeutic outcomes we need in patients. And Phage have been able to really kind of exemplify how hydrogels are a great platform for biologics, because we've taken these viruses that are diverse, come from different ecosystems, and then bring them into one platform that's able to co-deliver them and still maintain their viability. So it works as a strong proof of concept for the platform as well as being a viable product.
And the final hydrogels are powders? Is that
It
right?
looks
Like if you shipped them in the
I'll have
field?
to send you a picture, but usually they come in like a little tube, and there's a dry
Okay.
kind of white foam crystalline structure. It looks almost
Okay.
like almost like a sugar foam. You can see kind of the cryopreserving polysaccharides that are based inside of it. We use a variety of different sugars within there to help stabilize that matrix and keep those phage
Yeah.
stable. It's almost like a knockoff of amber, those saccharide matrices that are used to preserve essentially fossils for such long periods of time. We're cheating off of nature essentially to do a miniature version for viruses and more sensitive biologics. And so it's essentially just a white foam matrix. Once you add in the ionic buffer, it dissolves rapidly and blends in with that ionic buffer, and the cross-linking starts. So the physician has a certain amount of time to be able to then use a hydrogel for its indicated use and apply that to the patient, and then dispose of the final waste that's worked with.
So doing that sort of mixing in the field, you're finding is not an issue?
No, it's not very difficult because the ionic buffer is pretty small. We're looking at maybe 5 to 10 mils max. And that can be reduced further for a thicker hydrogel application. The goal is to just get the hydrogel into ionic conformation so it can set and it can do its job. But we've looked at other applications that don't require rehydration, like the inhalable version, where it's a dry powdered hydrogel. You don't need an ionic buffer. It rehydrates within the respiratory tract. And because of its biocompatibility, it's rapidly degraded and released and doesn't cause any respiratory issues. So it's really, really great that we can be as flexible as we can be with this technology.
Awesome. Yeah, it makes me wonder if hydrogels might be a suitable formulation platform for our compounds. Our compounds are proteins.
Yeah.
But they're I don't know what the molecular weight is of a phage, but our proteins are not gigantic.
Yeah, I'd assume that they're smaller than the phage. there's not a lot of proteins bigger than phage. There probably are. But nature's full of everything. But have y'all looked at mRNA delivery technology for your recombinant protein?
We have.
How has that been?
Yeah.
Have y'all done like liposomes and stuff for that kind of basis?
We're very interested in liposomes. We haven't done it yet, but there's some advantages to us for liposomal encapsulation.
Yeah.
mRNA is, I think, problematic.
Yeah.
just
Because you're
in terms of where it is located inside the cells
yeah.
and that kind of thing, and delivery. I mean, you can use, I guess, LNPs to deliver mRNAs.
Yeah.
That's what they do with the vaccines.
Yeah.
So
There's viral vectors
lipid
too,
nanoparticles.
adenoviral vectors and stuff, which can have like some
Yeah, viral vectors.
But again, that comes with issues because the patient might already be immune to the adenovirus. So yeah, it's a game of chess trying to figure this stuff out. Nothing's ever perfect, no matter what vaccine it is. We are such a diverse population that there's got to be some sort of adverse effect at some point. It's just keeping that risk to a minimum.
Yep. I think as with your platform, with the phages being specific to specific bacteria, we have a similar thing that our proteins are specific to specific cells, cell types, as well as viruses. So there's a lot of chess pieces, I guess you could say, in the game.
But it's really, you have like that inherent trophism. So like you don't have to worry about it being too off target. It'll at least target the necessary tissues that you needed to. So many drugs that we deal with nowadays have a lot of off-target effects and are indiscriminate with the way that they can really affect patients. We're seeing a lot of cases of antibiotic-associated toxicity coming up. One of my good friends was using a series of antibiotics for really bad, complicated upper respiratory infection. He developed tinnitus and neuropathy from dealing with those antibiotics. So they sometimes are just as toxic as what you're trying to treat, which is crazy, So being able to use biologics typically is a safer process for a lot of patients than these molecular drugs.
Yeah, one of my kids had sepsis a few years ago. It was very scary.
Yeah, absolutely.
Yeah, vancomycin and all that.
Yeah.
He recovered, fortunately, but it's very unpleasant experience.
Yeah, and
knowing that there's no good treatments.
Yeah, and we're trying to put stuff out there, but again, there's a lot of regulatory hurdles because again, they don't know how to classify and compare this because it's viruses versus drugs. How do you use a system that's built around drugs to regulate a virus that's a good virus? This is not a vaccine. This is something that's treating. So there's a lot of questions that need to be addressed from both a regulatory standpoint, but also need to be addressed to give patients options because they're essentially limiting outcomes because of a lack of understanding. And that can be difficult, but it's our job to educate,
Have you had your pre-IND yet?
No,
or is that
we're still holding off on that. We really want to make sure we have that strong animal trial data together so that we can really work with the FDA on that. We've been told that you really want to have a strong portfolio together for meeting with them. That way I can maximize that, limit the amount of meetings I have to have with them, and then take that feedback to our clinical partners. So again, trying to get that animal trial together and stuff. But we've got some great regulatory consultants that have been able to help us navigate some of that. Very expensive, but hey, they do a great job. And they know the field.
Yeah, we have a few of those too.
They can cost a lot, but it's great to have a defined pathway on how to move forward. So it provides a lot of clarity. And that's something great we can take to our investors and partners. And it gives us a kind of a direct path to work towards because there's a lot of if and ands when it comes to science. So having kind of a direct path from a professional can help a lot as well in that process.
Okay.
Well,
I guess that wraps it up for today. Thank you so much for your time.
Absolutely. Thank you so much, Rick. I appreciate it.
Great to chat.
Absolutely. Thanks for having me on. I really appreciate it. Let me know if you want to catch up anytime, okay?
Yeah, okay.
Perfect.
Thanks.
We hope you enjoyed this episode of Antiviral Frontiers. To hear more from the fascinating world of viruses, biotech, and medicine, please visit our website or find us on Spotify and YouTube and follow us to get future episodes.